Hi, everyone, and welcome to week 3 of your journey into the world of the unseen. Now, the big takeaway from this week is that bacteria are, well, sneaky little boogers. I know that's not proper scientific verbiage, but they are, for lack of a better phrase, sneaky little boogers. Over decades since the development of the very first antibiotic, you know that, as penicillin, that was back in 1941. Dozens of others have been developed, put on the market, and are currently in use in medical settings around the world. And yet, as brilliant as human science and medical researchers and engineers are, those sneaky little boogers, the bacteria, still seem to devise ways of skirting around our infinite attempts to control them, destroy them, and minimize their damaging effects upon us. And once again, the most unsophisticated organisms on the planet seem to have the upper hand over the most sophisticated organisms on the planet. And it makes me wonder, at least once again, who really is in charge down here. Well, down below I've provided a link to a short video from Science News, and in it you will see how bacteria are able to develop antibiotic resistance and confer that resistance onto their progeny in very short order. Now, when you recall that the generation time of E. coli is about 20 minutes, you can appreciate how fast things happen in the world of bacteria. And so, as you culture bacteria this week, pay particular attention on how fast those sneaky little boogers do their work. So, this week you will learn to culture bacteria using aseptic technique. More on that in a moment. And you'll learn about the effectiveness of various antibiotics, antiseptics, and disinfectants in controlling and inhibiting bacterial growth. So the housekeeping items for the week. Okay, so you should already have received your microbiology equipment kit, and if you haven't already cultured or reconstituted your bacterial cultures, you'll want to do that right away today. You have your initial discussion post due no later than Friday. That's this Friday, September 26, 11:59 p.m. GCU time. And your peer responses are due no later than Sunday, September 28, 11:59 p.m. GCU time. We're working on topic 3, methods of culture transfer, pure culture isolation, and antimicrobial testing. You have your pre-lab questions for labs 3 and 11 to do. Those are due no later than Sunday, once again, September 28, 11:59 p.m. And you want to start working, if you haven't already, on your 2 lab activities. You want to start them early because they need time to incubate. I made an announcement and posted instructions for doing just that on Friday, I think, or Saturday. Some of you hopefully have, I would hope that all of you have at least reconstituted your bacterial cultures. If not, you want to get those done right away, reconstitute them according to the instructions, and you want to do all 4 cultures at the same time. The instructions are located on the topic 3 page. You go to the right side vertical menu, scroll down near the bottom, you'll see the instruction manual from Carolina Biological Supply for the streak plate isolation and also the Kirby-Bauer assay. So you want to open those up, download those 2 instruction manuals, and then in there you'll find instructions for reconstituting your bacterial cultures. There's also a little instructional video there to help you along the way. And again, you want to do all 4 at the same time, and then you need to incubate those. Now, Prime conditions for bacterial incubation are warm, moist, and dark. The moisture comes by way of the septic broth, the soy broth, or nutrient agar broth. Your tubes may say one or the other. It's the same thing. You want the liquid, not the slant, not the agar slant, not the solid tubes, but the tubes with liquid in them. You want to make sure that you label each tube according to the organism that is in there, because you're going to have 4 different ones. You will place your tubes in your little rack here and incubate all 4 tubes together in a warm, dark place. Warm, dark, okay? I used to incubate mine on top of the refrigerator because it's at least warm up there. Cover them over with a towel to keep the light out. And but otherwise, I've done that before, and I've done. Interestingly, I have a lot of research equipment at my house, but I do not have an incubator, so I have to makeshift just like you all do. The other thing that I've done, I've put this with the 4 tubes in like a shoebox, and then shined a desk lamp on top. Shoebox for the darkness, the desk lamp for the heat, and then you come back in 24 hours, check them out. If your tubes are turbid, if you agitate them a little bit and they get cloudy, that means you have bacterial growth inside. That's a good thing. And then when you see turbidity within those tubes, then you can proceed to the next step, which are the agar plates. Same thing, follow directions. You incubate them in a dark, warm place. The moisture comes from the nutrient agar. Okay, so initially you should have 4 tubes incubating, and then once you get your cultures going, then you're going to end up with 3 plates, 3 agar plates, 1 streak plate. There's 2 organisms you have to mix together. Follow the instructions, Carolina instructions, and then in your and then for the Kirby-Bauer assay, that's where you use the little paper antibiotic discs. You should have 2 plates, one for E. coli, one for Staphylococcus epidermidis. Make sure that you label which plate is which plate. Also, make sure you put the date on there, because when you send me images, when you attach images of your plates to your assignment sheet that you then are going to submit, I need to see the date on there. Okay, so the date that you started the experiment, the date that you put the discs on there and you put them in the incubator, you need to see that date on there. And also for your streak plate, make sure the date is on there that I can see that. Okay, so that's that. My notes, as I see where some of the activity might be a little confusing, so I posted several videos to help you. I haven't, because those videos are embedded in there are some videos embedded in the literature from Carolina, but I will go ahead and post the videos that I put together a couple years ago that might help you as well. And so both of those assignments, the streak plate and the Kirby-Bauer assay, are ideally due no later than Sunday, September 28, 11:59 p.m. GCU time, but sometimes growth is a little bit slow, will be a little bit slow for some of you, so we have to sort of finagle the due dates, but just keep me posted as to how yours are going. Pay particular attention to due dates on all of your assignments. Proper credit can only be given for complete and timely submissions. Now, with regard to, before I close out, with regard to your discussion posts, many of you are doing a really fine job. I posted a sheet, a PDF, guidelines for discussion posts. You'll find it in class resources. Some of you need to look that over and look it over again. Your discussion posts need to be substantive. That's at least 150 words, and it must be supported by at least one authoritative reference. You can use your textbook, but if you use your textbook, you must also use a peer-reviewed journal article. When I show you how to find those, there's a video tutorial, again in class resources, on how to find those, okay? And then you must list the reference. Okay, you write your post, and then underneath you put references in bold print, or reference if it's only one in bold print, or references if it's more than one in bold print, and then underneath that you list your references according to the APA guidelines. But remember also that you must cite those references within the body of your text. We call those parenthetical citations because you put them in parentheses, okay? Follow APA guidelines on that. I have examples of all of that kind of thing in my guidelines for discussion posts, okay? So check that out. Make sure that if you don't do anything else, make sure you do a word count. And also I do scan for AI and plagiarism, so keep that in mind as well. All of your initial posts and your peer responses must be substantive. Your initial post is tier 1. Your responses to your peers are tier 2, and anything below tier 2. Tier 3 is when you reply to someone who has responded to your post. That does not have to be substantive. Only your initial post tier 1 and your peer responses tier 2. Only those have to be substantive, okay? All right. And I think that's it for now. Keep me posted on your lab activities. I will go ahead and upload those videos that I did, that I put together for students a couple of years ago. I'll put those in class resources. And other than that, I'm working on updating your grade books today. So don't worry about the red overdue on there. Okay, so as soon as I get yours done, it'll update and you'll be good to go. So keep me posted and have fun growing bacteria this week. Ciao.